Kingdom Monera then. Kingdom Monera, one of the 5 kingdoms. According to the Leaving Cert syllabus, we've got 5 kingdoms. We've got the plants, we've got the animals, we've got the fungi. Fungi are not plants at all, they don't photosynthesize, we'll look at them in another video. We have the Monera, and the Monera, as we'll see, is a kingdom consisting of only bacteria, so all the members of the Monera are bacteria, and all the bacteria belong in the kingdom Monera, so it's a fairly exclusive club. And then we have the leftovers. We've got 4 kingdoms here, and whatever's left in the living world that they couldn't fit into any other box, then they put them into this named the Protista. So they're a mixed bag, mostly unicellular, some multicellular, all pretty much small, mostly microscopic. But it's the Monera that we're focusing on today. The Monera are unusual and different, distinct from all the other 4 kingdoms. Just to be clear, then, this is the 5 kingdoms of the living world, and this one here, the Monera, are prokaryotic, and we'll just have a look and see what that means. So the prokaryotic organisms, the bacteria, all have are distinct from or different from other organisms because they have no nucleus. They've got no nucleus, and they also have what's absent from them is any kind of membrane-bound organelles. So what we're talking about there is they like things like mitochondria, chloroplasts, and obviously no nucleus. They don't have any of those kind of organelles. They belong to the other 4 kingdoms. So these 4 kingdoms here, these 4 kingdoms here then are what are known as eukaryotic. So these are the eukaryotes. Eukaryotic. Okay, they're the eukaryotes. E-U-K-A-R-Y-O-T-I-C. The eukaryotic. So prokaryotes, no nucleus or other membrane-bound organelles. The eukaryotes have a true nucleus. So let's look at the bacteria, the structure of a bacterial cell. I'll just do a quick drawing of it. So the bacteria come in various shapes and sizes. We'll look at the different kinds of shapes at the moment. Let's just try and look at a general structure of a bacterium then. On the very outside you would have occasionally you have something called an outer layer, which could be a capsule. Not all bacteria have it. It could be a slime layer. So some bacteria will have an outer layer, like I said. All will have, or almost all will have, what's called a cell wall. So like plants and like fungi, bacterial cells do have a cell wall, but it's made of something different to either plants or fungi. So this is this layer here. We'll just label that. Let's see, we'll go for this color here. This then would be a capsule or a slime layer, and they could be for attachment, they could be for protection, and they could be for, like I said, adhesion or attachment. Often associated with pathogens then, these capsules or slime layers. The next layer in will be a cell wall, and as we said, this cell wall is not made of cellulose like plant cell walls, not made of chitin either, which is what the fungal cell wall is made of. Inside that cell wall then you would also see, I will just draw it in a different color just to be clear, you would have the cell membrane. So cell membrane then just inside the cell wall, as you'd expect. And that would be our cell membrane there. So cell membrane. Okay, apart from that, we're looking at then, these are the structures on the outside. On the inside then, we are looking at a situation where, like I said, there's no nucleus, but there is one single chromosome. So chromosome will be a large coiled chromosome. This will be one large circular structure, and that is not contained in any membrane-bound nucleus or anything. That it's just part of this floating around in the cytosol. Apart from this DNA here, what keeps or sets the bacteria aside is that they often have these small circular pieces of DNA. Often they'd have more than one. Different sizes relative to each other, but always much smaller than the chromosome. So you might have one or two, or it could be multiple, it could be up to 12 or even more of these small single-stranded, sorry, not single-stranded, small circular pieces of DNA. Could be single-stranded, some are single, some are double-stranded. But they are all known as what we call plasmids. And we'll see later that plasmids often encode things like antibiotic resistance or the ability to use a certain kind of substrate. Plasmids can be passed in between bacteria as well fairly efficiently, and so that's how they increase variation within a population. They swap these around the place. Some bacteria are fairly good at it, and so this is how they might share genetic information and increase variation. Because, as we'll see later, bacteria don't have the option of sexual reproduction to increase variation in their populations, so they rely on mutations and the swapping or the exchange of these plasmids. So, apart from that, you will find, as you will find in lots of other organisms, you will find that you have ribosomes. So, little ribosomal protein synthesis factories, protein making factories scattered around the cell. In another video we'll look at protein synthesis and how these ribosomes work. So you'd expect to see those if you had a light microscope. But you'd also expect to see on the outside, I should have put this in, I'll just put it in a slightly different colour just for variation, is that we've got, often you have a flagellum, which is a long, and sometimes there's a series of them. They are long structures associated with movement, so they're a bit look like a tail. They don't operate like a tail. They operate more like a propeller. Actually, they've got a kind of propeller sort of action. So that is the flagellum, and that's for movement. Flagellum. Flagellum is singular. Bacteria have flagella, plural. Okay. So just the last label then we were going to label was ribosomes. There's another ribosome. Ribosomes. Okay. So let's just continue on. What we'd also look at then just briefly is how bacteria undergo reproduction. So if we have one bacterial cell here, and inside that bacterial cell we've got a chromosome, what the bacterium will do is it will, just before it replicates, it will elongate itself, so it will become longer. It will duplicate the chromosome, so the chromosome will get copied into another chromosome, just an identical copy of it. And the reason, of course, that it needs to do that is because it's going to then begin to split in two, and it does this in a mode that we call binary fission. So it's binary fission literally just means splitting in two. So each of the new daughter cells, or each of the new progeny of the cells, will then get its own individual chromosome. Of course, any plasmids that are contained in the cells, they will have got replicated as well. And in the end, then this cell will split in two, and you get two bacterial cells excuse me, for the price of one. So that's what we call binary fission. Binary fission. Binary. So it's a bit of a fudge to call this binary. Binary fission just means splitting in two, and it's also applied to yeast, for example. When we look at yeast, remember though that yeast undergo mitosis, which is nuclear division first. There's no nuclear division involved with bacteria because there's no nucleus. So prokaryotes don't have this complicated process of mitosis that has to come before cell division. So this is a much more, if you like, streamlined or simplified way of reproducing. But it's asexual. Okay, so this is asexual reproduction. There's only one parent involved. This is a parent. The offspring will be identical to the parent and to each other, assuming that there have been no major mutation events, and there's always going to be some mutation events. So as these generations go on, mutations can build up within a population. I should also say that this process can happen just from beginning to end. It's possible with some bacteria this can happen every 20 minutes. So that's a very short generation time, and it means that a population of bacteria from just one cell can grow to millions in a very short space of time at all. And if you look at how many are formed in the space of 24 hours, it's a serious number of bacteria. So why is it that that the place isn't, the world isn't heaving with metric tons of bacteria? Well, we'll look at that in a second. Just to look at the shapes then, if we move on, shapes of bacteria, they come in different kinds of shapes. Generally speaking, we're looking at rod-shaped bacteria, and that's sort of self-explanatory. They might be this shape. They're sometimes known as bacilli. Bacilli is another bacteriological word for rod-shaped. Sometimes they're spheres, so that would be one kind of shape. You have spheres. Spheres are also known as cocci. So you have streptococcus or staphylococcus. They're commonly known bacteria, and they're spheres. Often you'll find them associated with in tetrads or in pairs, sometimes as long chains of spheres or cocci. So these are the rods and the cocci. Most bacteria come into those two categories, but there's some other shapes as well. You might have spiral-shaped bacteria, so you would have this sort of a thing, where they would be spiral-shaped. So some bacteria like that, spirillum would be one type of bacteria that has that kind of shape. Or you also have comma-shaped, which are this sort of a shape, like a kidney bean sort of a shape perhaps. Sometimes not as shaped like that, but also like this. And then these are the comma-shaped bacteria. The most commonly known one, like comma-shaped bacteria, is Vibrio cholerae, and that was responsible for, as the name suggests, responsible for the cholera epidemics that occur in the world still in developing countries. In the underdeveloped countries, cholera is still a big issue. So that's the shapes of bacteria. So let's just look at then the different modes of bacterial nutrition. Okay, so if we look at nutrition in bacteria, we've got the basic groupings are, we've got some bacteria are heterotrophs, heterotrophic. Heterotrophic means that they are relying on the consumption of other organisms to get their nutrition. So they eat other living other organisms of some sort. They can be in two kinds of heterotrophs. They could be saprophytes. Saprophytes live on dead organic material, so dead organisms or products of organisms, parts of organisms, as opposed to the symbionts. And the symbionts, symbiotic bacteria or symbiotic organisms in general, come in three different possible modes as well. So we have parasites, we have the mutualists, and we have the commensals. Now, just to go through these, because I think this is important, not just in the bacteria chapter, but in general. Symbiosis is a close relationship, or an intimate relationship between two organisms, a feeding relationship in which at least one organism gains. So at least one gains. At least one organism benefits, okay, or gains something out of the transaction. Now, in the case of parasites, just so we are clear, in the case of parasites, one is harmed and one benefits, obviously, because at least one has to gain. So this one, in parasites, one is harmed. When it comes to the mutualist, both benefit. Oops, sorry, I just didn't do that. One is harmed. In the case of mutualists, they both benefit. Okay. And in the case of commensals, one benefits, as we said, which is the case for all symbionts, at least one gains, but the other one is, there's no effect on the other one. So in the case of the other one, it neither is harmed nor benefits. So neither the other, sorry, let me just do that again. Other is unharmed, but nor benefits. Nor benefits. Okay. So that's the breakdown. I just want to put in some detail there about symbionts. So symbiosis again, close relationship between two organisms where at least one gains. Parasites is one kind of symbiont, and it's the relationship where one of the organisms is harmed and the other one obviously benefits from that. If both benefit, we call it a mutualistic relationship, and if it's a commensal relationship, we are looking at a situation where one of the organisms is gaining and the other one is neither harmed nor benefits. Okay, so other than that, apart from the heterotrophs, going back to one of the two major ways that bacteria can gain their nutrition, we have the autotrophs. Now, we should know that plants are the obvious autotrophs. Autotrophs are organisms that can make their own food from simple inorganic raw materials. Now, plants do that with water and carbon dioxide. So mustn't forget this about plants, is that they do this amazing thing where they stitch together water and carbon dioxide from the air, and they make glucose from that using the light, the energy from sunlight. But there's other ways of doing the same sort of trick. We're using simple inorganic raw materials and making food out of it. So one way is the photosynthetic ones, which we know about. So and bacteria, some bacteria are photosynthetic. Bear in mind they don't have chloroplasts because they're prokaryotes, but they can make energy from they can use light to make food. And then there's the chemosynthetic bacteria, and these are strange creatures. So the chemosynthetic bacteria, an example of those, these are organisms that use the energy from chemical reactions to make their food. So this is about using chemical reactions. And the example would be, in our course, the example we have is the nitrifying bacteria. So just to put that in there, then these are the nitrifiers, the nitrifying bacteria, and we see those when we look at the nitrogen cycle, which I'll do in another video. Okay, so that's the overview of bacterial nutrition. Just to move on then. So let's look at bacterial growth. There's factors affecting bacteria. Just to have a quick look at those. So factors affecting bacterial growth. If we look at bacterial growth, we're looking at factors like temperature. I think that would be fairly obvious. Earlier on we were wondering why it is that the bacteria haven't just taken over the planet and the whole, if they're able to divide, reproduce every 20 minutes, you'll quickly see that it's exponential growth. If the conditions are right, then they should be able to make tons of themselves, literally metric tons of themselves within just a few hours. But because these things are limiting, because temperatures never or rarely ideal for these bacteria, they're able to reproduce every 20 minutes. But it's not just about temperature, it's also about acidity levels, pH of the environment, it's about oxygen levels. Some bacteria are aerobic and they require oxygen. They're sort of strict anaerobes. Some are what we call facultative. So it could be that the aerobes are obligate. In other words, they have to have oxygen. They must have O2. That means they're obligate aerobes. Or they could be facultative, which means that they can switch. They're fine with their oxygen around. They'll use aerobic respiration. But if there's no oxygen around, then they will be able to manage without. They can respire with or without. Okay, they can respire without O2. So those are the aerobes. We do have anaerobes as well, and you could say that these are the facultative aerobes or facultative anaerobes, whichever you want to call them. Either way, that they can do with or without. But you do have obligate aerobes, or anaerobes rather, are strict anaerobes. They are strict in the sense that they must have, they can only respire in the absence of oxygen. So they can only respire in the absence of oxygen. So if there's oxygen present, for these guys, they'll die. So examples of that are Clostridium botulinum, which we'll look at when we look at some examples in a minute. Okay, so that's bacterial growth. Let's just look at a situation where we might have a bioreactor, and a bioreactor would just be a large purpose-built vessel, usually a steel vessel or steel and glass. That is, it's perfectly sealable, so it can be sterilized. The interior, the inside of it, can be sterilized and filled with sterile nutrient medium for the bacteria to grow in. And then the bacteria will be inoculated in using the aseptic technique, which we'll look at when we look at our experiments in microbiology in another video. And we'll just go through this batch growth curve then. So if we just draw, let's see if I can do better than that. Let's see. So if we have a batch curve, so we'll draw these axes here. Should draw that out to about there, and this is going to be on this side. We're going to have this is going to be the number of bacteria cells. So number of bacterial cells, and here we have time. So we'll look at what happens with the growth of bacteria over time. Try and do this in stages, and look at the stages one by one. So the first stage will be, just undo that. The first stage will be a slow to start kind of a stage, and that first stage is what's known as the lag stage. So I'll just put in some labels here. This stage here, stage A is the lag stage. of the lag phase. So the lag phase is characterized by cells that are only growing slowly, they're only dividing slowly, and the reason for this is that they're having to adapt to their new nutritional environment. Okay, so there's a delay or a lag as the genes that are necessary for the production of enzymes, the enzymes that they require to be able to deal with the new food environment that they find themselves in. There's a delay or a lag as those genes are switched on or expressed. So we'll look at gene expression again in another video, but just for here, just so we know that this is what the delay is caused by as the bacteria get used to the new environment. Okay, this stage then is followed by the log phase, and the log phase is characterized by an exponential growth. So this period here where you have this logarithmic kind of a shape to the curve, this is known as the log phase for that reason. So stage B then is the log phase, or logarithmic, sometimes known as the exponential phase. Okay, and the reason it's called this, like I said, is because growth is exponential. Conditions are favorable, optimal if you like, they're perfect for the growth of the bacterium. Growth is really fast, metabolism is very high, and the rate of cell division is at its highest. Now, this is fine, this is going to happen because there's plenty of food around, conditions are all perfect, temperature's perfect, pH levels, there's no waste in the environment, there's no competition. That won't last very long, obviously, because bacteria are going to find themselves in a situation then where the buildup of waste starts to become an issue. There's more competition, and the environment around them is changing. So as the waste is produced, that could be could change the pH of the medium, and food is being used up. So even though the temperature is the same, other limiting factors kick in, and this stage, stage C then, is what we call the stationary phase. So C is the stationary phase. The reason that this is flat here is not because there's no growth at all. And remember, this is just the number of bacterial cells in the population. So the population is actually staying steady here. The numbers in the reactor are the same. And what happens is the reason for that is because the number of cells that are dying is equal to the number of cells that are reproducing. So the number of new cells, if you like. Now in this situation, then, like we said, conditions are becoming less than optimal. There's competition with other bacteria, for nutrients, for oxygen. Waste is becoming an issue. It's building up, and it's becoming increasingly toxic for the bacteria to survive. So this is why we have this flat part of the curve, which is called the stationary phase. Now, as things worsen, then in the environment, then obviously that's going to have a detrimental effect on the bacteria. And so quite quickly then we see this decline, which is the next stage. So we call it the decline or the death phase. The decline phase or the death phase. And what's happening here is that levels of toxicity are just now becoming too much, and bacteria are finding it increasingly hard to survive. The number that are dying is far outweighs the number that are able to reproduce, and it's just a further extension of the situation that we talked about here, where things are getting worse in terms of buildup of toxins and availability of food and change in pH, buildup of waste. Okay. Finally then, and it only applies to a few bacteria. Most bacteria then will just, in this situation, the population will completely die out. Don't know what happened just there. Let me just put that back in here. This is time. We said time here. So as time goes on, most bacteria, this curve will just reach a bottom, the number of live or viable bacteria cells will be reduced to none. There are some bacteria, though, that have a trick up their sleeve, and they're able to survive in situations like this. So this is what we call the survival phase. And it just means that on the curve, what it looks like is that a few bacteria will be able to, as the name suggests, will be able to survive. So in very low levels then, you'll be able to see this background number of viable cells. And the only reason why that might happen in this sort of an environment where things become very, very toxic and hazardous for bacterium is that there are some bacteria are able to produce structures known as endospores, which we'll look at. So endospore formers are bacteria that can produce these little survival pods, if you like, and are able to survive very hazardous conditions.